Hello, dear students. All of you, welcome back in our third session on the course Ethnobotany. Credit number first. From that credit number first, the unit number 4, the future perspective. The topic, the phytochemical analysis of active principles and their pharmacological uses. So, firstly, I introduce myself. I am Dr. Mukta Bhamre, Assistant Professor at Department of Botany, KTHM College, Nasik. The session agenda of today's lecture is the introduction, the concept of active principle, phytochemical extraction methods, phytochemical screening or analysis, and the phytochemical analysis of some medicinal plants. All these points we will cover in our today's session. So the first I am focusing here the introduction. So the preliminary screening of phytochemicals is the valuable step in the detection of the bioactive principle or compound present in the medicinal plant, and the subsequent may lead to drug discovery and the further drug development process. So plants are recognized in their pharmaceutical industries for their broad structural diversity as well as their wide range of pharmaceutical activities. These phytochemicals are derived from the various plant parts such as the leaves, flowers, then seed, bark, root, and pulp. These phytochemicals are used as a source of direct medicinal agents. Means from ancient period of time, the different medicinal plants were used as a source of medicine. Then the phytochemical screening should include the identification of active substances, then extraction with a suitable solvent system, purification and the characterization of active ingredient for pharmaceutical purposes. In this method, the aqueous and various organic extract are prepared from those plant samples that are reservoir of the various secondary metabolites. Means from that, using the different extraction method, from that different extraction method here, preparation of the various solvent, preparation of various extract by using the solvents, various polar and non-polar solvent, and these solvent they specifically leach out the variety of secondary metabolites present within that plant part. The plant extract are then analyzed for the presence of secondary metabolites like as alkaloids, terpenes, and flavonoids, tannin, like that. Then next, I'm focusing here the concept of active principle. What is the active principle? The active principle of the drug are the chemical constituents that are responsible for the therapeutic effect. Active principle, it is nothing but the active ingredient or active constituent or active entities present within that plant parts or plant body, and that active principle is having a very important role in a physical physiological processes as well as in it also shows the various biological activities and it shows the therapeutic effect. In a medicinal plant, the synthesis of variety of secondary metabolites like as alkaloids, saponins, phenolic compounds, terpenoids, and flavonoids, then phytosterol shows the therapeutic effect and they having a special physiological action. Such chemical entities are nothing but the active principle. So the active principle, they are having a two different types. One is the inert constituent or inert active principle, and second is a active principle. So first I'm focusing on the inert active constituent. Means here that inert active constituent they do not having a the therapeutic effect, means it's having but it is a very little bit, but they are the major part of the structural component of the plant body. Here the example like as a cellulase, lignin, suberin, cutin. Okay, all these are the important constituent or important structural unit of the cell wall of the plant. Then starch, it is a reserve food material present within the plant body. And albuminoids, it is also present within the plant body. So these are specifically primary metabolites. Okay, they do not having any significant therapeutic effect. Then second, I am focusing here the active principle, means it is nothing but the active constituent or active ingredient present within the plant extract. They specifically shows the therapeutic effect, means they specifically cure the various diseases, and they are used as a medicine from ancient period of time. So this active principle, they specifically it is in the form of alkaloids, then saponins, then phenolic compounds, terpenoids, flavonoids, and the phytosterol. So the next I am focusing here the phytochemical extraction method. So before begin with the phytochemical analysis, here it is one of the most important step, that is the extraction of phytochemical. How we can extract the phytochemicals from the various plant parts. So the first I am here the emphasize the extraction. So what is the extraction? Extraction, it is a nothing but the separation of the medicinally active portion or active principles of the plant tissues using the selective solvent through the standard process. So the basic parameter that influence the quality of extraction, specifically the plant part used as a starting material, means which plant part we specifically used for the extraction, means here the stem, root, leaves, seeds, fruit, pulp, bark, like that. Then the second important parameter, it is a solvent system. So there are various solvents are polar and non-polar solvents. Okay, but here the polarity it's determine the extraction process, means some that secondary metabolite they are extract out in the polar solvent system, and some are extracted out in a non-polar solvent system. And the last one, the parameter influence, that is the extraction procedure, means which specific extraction method we will choose for the extraction of phytochemicals. So here I'm focusing the choice of the solvents. So the successful determination of biologically active component depends on the types of solvent used in the extraction process. So the properties of the good solvent in the plant extraction, they have a low toxicity, then the easy it is of evaporation at a low heat or low temperature, then promotion of rapid physiological absorption of the extract, and the last one, that is the preservative action. These are the important properties of the good solvent. Then the various factor affects the choice of the solvent system. Here that factors means quantity or quantity of phytochemicals can be extracted, then the rate of extraction, then diversity of different inhibitory compound extracted, then the easy of the subsequent handling of the extract, toxicity of the solvent in the bioassay process, and the potential health hazard of the extractant. These are the some factors, they specifically affect the choice of the solvent system. Then the next I am focusing here the solvent used for active component extraction. So in the field of the chemistry, that is various solvent systems are present, but we choose the solvent according to our interest, according to our choice, means which active component we specifically extracted from that plant material, then according to that we can use the solvent system. So the water, it is one of the best universal solvent. This water, they specifically extracted the anthocyanin pigment, then the starch, tannin, saponins, terpenoids, then polypeptides and lectin. Okay, When we extract these phytochemicals, then we choose the water as a solvent. Then the ethanol, it is specifically used for the extraction of tannins, polyphenolic compounds, then polyacetylene, then flavins, then terpenoids, sterols, and alkaloids. Then methanol, it is specifically used for the extraction process of anthocyanin pigments, then terpenoids, saponin, tannins, then xylines, then toterol, then cuisinoids, then lactones, flavonoids, phenols, and polyphenols. Then chloroform, it is a use for the extraction of terpenoids and flavonoids from that plant material. Then ether, it is a use for the extraction of alkaloid, terpenoids, then curcumins and fatty acid. And the acetone, it is a use for the extraction of phenol or phenolic compounds and flavonoid. So this is the solvent system, or we use the different solvent for the active component extraction processes. Then next I am focusing here the general techniques. Means here the phytochemical extraction method, here the general techniques of the medicinal plant extraction. So there is various techniques they specifically use for the extraction of phytochemicals. Specifically the plant tissue homogenization process, then maceration process, then infusion, then percoration process, then digestion, then decoction, Soxhlet extraction method, it is a hot continuous extraction. Then sonication, it is ultrasonication method. All these are the technique they specifically use for the medicinal plant extraction, the extraction of crude drugs from the medicinal plants. Then the phytochemical extraction method, I am focusing here the plant tissue homogenization. So the first, we collect the plant sample from the field or from the forest or any natural resources. Then here, collected sample, they specifically wash with the tap water. Then it is a dried means it is sun dried, or it is a wet, or we can use a fresh plant material, plant parts. Then it is a grind in the blender to the fine particles along with the certain quantity of the solvent. Then shake vigorously for 5 to 10 minutes, or it is left for the 20 hours after the which extract is filtrate. Then after that, the filtrate is dried under the reduced pressure and redissolved in the solvent or centrifuge for the clarification of the extract. Then maceration, it is another technique for the phytochemical extraction. The whole or there is a coarsely powder crude drugs is placed in the stopped container with a solvent system, allowed to stand at the room temperature for the period at least 3 days with a frequently agitation until the soluble matter get dissolved. The mixture, then it is a strain, the means the damp solid material is pressed, and the combined liquid is a clarified filtration, or there is a decation after the standing. Then infusion, it is also another method for the extraction of phytochemical. Fresh infusions are prepared by the macerating the crude drugs for a short period of time with the cold or with the boiling water, and these are the dilute solution of the readily soluble constituent of the crude drugs. Then digestion, it is also another method for the extraction of phytochemicals. Form of maceration in which the gentle heat is used during the process of extraction, used when the moderately elevated temperature is not objectionable. Then the solvent efficiently of the menstruum is thereby increased. Then there is a next method, decoction. The crude drugs is boiled in a specific volume of water, 1:4 proportion for a definite time period. Then volume is reduced in a 1/4 quantity, the original one. And then it is a cool down and the strain, or it is a filter, and it is a suitable for the extracting the water soluble or the heat stable constituent. And typically it is a used in the preparation of Ayurvedic extract in a Ayurvedas. Then the percoration, the use mostly frequently to extract the active ingredient in the preparation of the fluid extract. The solid ingredients are moistened with the appropriate amount of the specific menstruum, allowed to stand for approximately 4 hours in as closed container. After the stand time, the mass is packed and the stop of the percolator is closed and the mix is allowed to macerate in the closed percolator for 24 hours. Then additional menstruum is added is required until the percolate measure about the 3/4 of the required volume of the finished product. The mark is the press and express the liquid in added to the percolate, and sufficient menstruum is added to produce a required volume, and the mixture it is properly mixed and liquid is clarified by filtration or by the standing followed by the decaying. So the next important method for the plant extraction, it is a widely used, it is a stocklet extraction method. It is also called the hot continuous extraction method. The fine ground crude drugs is placed in the porous bag or the thumbnail. It is made of the strong filter paper which is placed in the stocklet apparatus. After that, the extracting solvent, it is in the flask, is heated at a specific heating point or boiling point of that solvent is adjusted, and the vapors condensed in the condenser. That collected vapors, then it is a cool down. The condensed extracted drip into the thumbnail and containing the crude drugs. When the level of the liquid in the chamber rises to the top of the siphon tube, the liquid content of the chamber siphon into the flask, and then this process is a continuous and it is carried out until a drop of solvent from the siphon tube does not leave the residue when it's get evaporated. So see in the diagram, there is a stocklet apparatus is there is a upper part, there is condenser is there, then thumbnail portion is there, and lower there is a boiling flask is there, round bottom flask. Then next there is a sonication. It is also the another important process for phytochemical extraction. It is also called the ultrasound extraction. It's involved the use of the thousands with frequencies ranging from 20 kilohertz to 2000 kilohertz, increase the permeability of the cell wall and produce the cavitation. Then the disadvantage is the very deleterious effect of the ultrasound energy on the active constituent of the medicinal plant to the formation of the free radical, and it is a consequently undesirable changes takes place in the drugs molecule. The variation in extraction methods, the length of the extraction period, it is also determine the process of the extraction, means the length of extraction period like as 1 hour, then 3 hour, 4 hour, or the 12 hour, 24 hour, 48 hours. This time period, it is also means influence the extraction of the phytochemicals in a crude extract. Then the solvent use, the specifically I'm focusing in a last slide. The some phytochemicals, they are extracted in a polar solvent system and some are extracted in a non-polar solvent system. So the selection of the solvent system, it is also most important. For the polar solvent we used for the extraction of polar phytochemicals constituent. Then the pH of the solvent system means it is a pH, it is a acidic pH, then it is a acidic pH, basic pH, or neutral pH. The pH range is also badly affects the extraction method. Then temperature, the different solvent, they have a different boiling point. For example, the water boiling point is 100 degrees Celsius, and the ethanol, methanol, petroleum ether, it is a boiling point above 30. Means here the boiling point of the solvent, they also means important, And the temperature, it is also plays an important role in the phytochemical extraction method. Then particle size means the particle size of the plant tissue. If the particle size is larger, then also affect the extraction process. And the solvent to sample ratio means which solvent system we used, which plant material or the quantity of the plant material, then we can choose the appropriate solvent sample ratio. So these are the variation in the extraction method. Then next I am focusing here the phytochemical screening method or the phytochemical analysis. So here the qualitative analysis focused here the phytochemical, then the test performed, reagent used, and the end point means it is a positive results. The phytochemical alkaloid within the crude drugs, detection of alkaloid, presence of alkaloid. For that, the Dragnofol test was carried out. The Dragnofol reagent used and the prominent yellow PPT, they indicate that the presence of alkaloid within that crude extract. Then the Wagner test, they use the Wagner reagent, and the end point of this test, reddish brown PPT was produced. It indicates that the presence of alkaloid. Meyer test, the 1% of hydrochloric acid and Meyer reagent were used, then turbid extract is obtained. It shows that the presence of alkaloid within that plant sample. Then the flavonoid, for the detection of flavonoid, the ammonia test and the sodium hydroxide test were carried out. The 1% ammonia solution used and the yellow color, the positive result indicate the yellow color means it is the presence of that flavonoid within that sample. Then sodium hydroxide test, 20% sodium hydroxide and hydrochloric acid, the yellow color turns to colorless means it is indicate that the presence of flavonoid within that crude extract. Then the presence of tannin carried out with the help of the ferric chloride test. 5% ferric chloride reagent as used, and the end point was the blue-black to blue-green color formation. It shows the presence of tannin within that crude extract. Then phenolic compound detection, the gelatin test was carried out. The 1% gelatin solution containing the 10% NaCl. Here, the end point, the white precipitation obtained, it indicates that the presence of phenolic compound within that crude extract. Then the saponin test, here specifically the lead acetate test and foam test were performed. The 10% of lead acetate used as a reagent. Then bulky white PPT were produced. They indicate that the presence of saponins. Then the another foam test was also routinely used. Means 20 ml distilled water. It is a presence of the fourth mix vigorously, and then there is a boiling of that water. There is a forming the foam on the surface of the boiling water. Then it's indicate that there is a presence of saponin within that crude extract. Then the detection of phytochemical like as a terpenoid, the Salkowski test were used. The 0.5 ml chloroform, it's a reddish brown coloration at the interference. It shows the presence of the terpenoids within the crude plant extract. Then the detection of carbohydrate, the Molish and Fehling tests were performed. The concentrated HCl, then violet ring appear, then at the middle, it's indicate that the presence of carbohydrate within that sample. Then the concentrated HCl used. Then here it is a turning concentrated HCl along with the magnesium. Then it shows the yellowish and the brackish red color precipitation was found. It indicates the presence of carbohydrate within that crude drugs. then crude extract, then protein. For detection of the protein, the biuret test was used. The 4% sodium hydroxide and 1% copper sulfate. Then the end point of this test, the violet or pink color developed. They indicate that the presence of protein within that crude extract. Then the detection of various glycoside. There's the legal test and the Kilani test were performed. For the legal test, the pyridine and nitroprusside used, and the end point here, the pink red color were developed. Pink color indicate that is the presence of the glycoside. And the second is the Keller-Kilani test. The glacial acetic acid and 5% ferric chloride were used, and the reddish brown and bluish green color developed. It indicates that the presence of glycosides. Then the next, I'm here focusing the quantitative analysis. I'm here taking the total phenol content and total flavonoid content. For the quantification, here the quantification carried out determination by the Folin Kai-Calcu assay method, and here the instrument use the UV visible spectrophotometer. Then absorbance measured at 760 nanometer. Then here the expressed as a gallic acid equivalent in the milligram per gram of the fresh leaf. When phenolic compound, the gallic acid used as a standard. Then for the total flavonoid content determination, it is determined by the colorimetric method, Yan et al. In 2009. Instrument used UV visible spectrophotometer and absorbance measure at the lambda 415 nanometer and express as mg routine equivalent per gram of the fresh leaf. This is the quantitative analysis of the phenolic compound and flavonoid content within the crude plant extracts. Then the next here, the phytochemical analysis of some medicinal plant, how the phytochemical analysis takes place. See the phytochemical analysis of some medicinal plant. So to carry out the quantitative and qualitative phytochemical analysis of the selected medicinal plant, here the methodology, here the selected some medicinal plant like as a Bryophyllum pinnatum leaves, then the Ipomia aquitessa leaves, then Olendra corymbosa whole plant, then Tinospora cordifolia, we call the goodwill, use the leaves and stem. Then Ricinus cuminis errand, the root used. Then Terminalia bellerica leaves and Xanthemum leaves. These leaves, root, then stem, all these are taken as a source for the crude, taking as a medicinal plant source part. Then the preparation of extract. Hot water extract and solvent extract, there is a two methods are there. The 5 gram of the dried fine powder of the plant material, and these powder mixed with the 200 ml distilled water, then heated on the hot plate with a continuously turning at 30 to 40 degree for 20 minutes, and after that the filtrate throughout the filter paper, and the water extract water extract formation. Then solvent extraction method, 20 gram of powder, plant material powder packed into the thumbbell and extracted with the 250 ml of the solvent. We can use any solvent system, methanol, ethanol, alcohol, then petroleum ether, chloroform, hexane, like that. And extraction continuously for 24 hours or till the solvent in the siphon tube of the extractor, it become a colorless. Then it is kept on a hot plate and heated at 30 to 40 degree. Then evaporation of the solvent takes place. Then phytochemical analysis, the qualitative and quantitative. The qualitative analysis here, I'm already focused in the previous slides. The protein content, it is a detection of protein content by Millen's and inherent test were performed. Carbohydrate detection, fillings, then Bentic reagent, Molish reagent, iodine test, then phenols and tannin detection, the ferric chloride test, then flavonoid detection, the Shindoda and alkalines reagent test, then detection of saffron in the foam test, then glycoside, there is a Lieberman's, then Kilani test, then detection of steroid, terpenoids, alkaloid. For the detection of alkaloid, Myers reagent test is there, Wagner's reagent test is there. It is for the qualitative, means preliminary screening, or the detection, there is a presence or absence of this phytochemical. Then if that phytochemical present within that crude extract, then we go next for the quantitative analysis. In the quantitative analysis, we specifically measure the total phenolic content, total flavonoid content, total tannin content, total terpenoids content, okay, then total alkaloid content, like that. Then next, so the result and discussion, the phytochemical constituent of the medicinal plant. So there is a 5 to 6 plants are there. Then see the protein, carbohydrate, phenolic compounds, tannins, flavonoid, saponin, sterols, then glycosides, terpenoids, and alkaloid. So the plus sign, it's indicate the presence of that compound within that extract, and minus sign, it's indicate the absence of that compound. So the bryophyllum, within that there is a presence of all protein, carbohydrate, phenol, flavonoids, saponin, glycosides, steroids, terpenoid, and alkaloid. So Ipomia, they also have all these phytochemical. Then Olendra, it's also having all. Then Racinus, it's only shows the absence of alkaloid test, it's the negative test. Then Terminalia bellerica, it also shows the absence of alkaloid. Then Tinospora cordifolia, it shows the presence of all the phytochemicals like protein, carbohydrate, phenol, flavonoids, alkaloids, saponins, sterols, terpenoids. Then xanthium, it shows the absence of steroid and terpenoid. And alkaloid, saponins, flavonoid, phenolic compound, carbohydrate, proteins, they show the presence of these phytochemicals. Then see the phytochemical analysis of some medicinal plant. Here the total phenolic content. See first graph indicate that there is a total phenolic content mg per gram of extract. The xanthemum, it shows the more phenolic content as compared to the bryophyllum. Okay, the lowest, it's having a 11.6 in a Olendra, and highest it is a presence in a xanthium, 71.6. Then the second graph, it is for the total flavonoid content. So the total flavonoid content, it is a more in 42.8 mg gram of plant extract. It is found in a Terminalia, and the lowest, it is a 4.4. It is found in a Olendra, means it is for the total phenolic content in the different medicinal plants. It is a phytochemical analysis. Then see the conclusion of this preliminary phytochemical analysis or screening of the phytochemicals. So this result revealed that the extract from this plant can be used as a good source of drugs, and further work should be carried out to isolation, purification, and characterization the active constituent responsible for the activity of this plant. So the today's session, we see here, we already focused the various phytochemical extraction method, what is the active principle, then there is a phytochemical screening methods. So on that, the student assignment is given here. So what is the active principle, or define active principle? Then question number 2, define extraction. Then question number 3, analyze the different method of phytochemical extraction. Then what is a maceration, and analyze the different types of phytochemicals. So all these 5 questions given this student assignment section. Then see in the next session, we will focus the phytochemical analysis of the various plant parts, and specifically the alkaloid, tannin, phenol, and we focus the therapeutic uses of these crude drugs in the field of medicine. We will focus all these points in our next session. Thank you.